SARS-CoV-2 first emerged in China in late 2019 and has since spread rapidly around the world. Trying to understand why the severity of the disease can be so different between individuals is necessary to the ongoing management of the disease. It is my pleasure to introduce Dr. Lee Murphy, the manager of the Clinical Research Facility at the University of Edinburgh in Scotland. In this webinar, Dr. Murphy will present how a multi-omic approach can help shed light on the host response to the SARS-CoV-2 infection, from the diversity of the sample needed to the different aspects of the analysis themselves. Dr. Murphy, the floor is yours.
Thank you. So I'm going to talk about two projects that we're working on over the last 3 months during this pandemic that uses a genomics approach to try and understand the differences in disease progression in people who are infected with SARS-CoV-2. First of all, my disclaimers and disclosures. So these are my views and not those necessarily of Illumina. So I work at the Edinburgh Clinical Research Facility, which is a joint facility between the University of Edinburgh and the NHS Lothian, our local health provider. And here we're specialising in clinical research. I head the genetics lab, and we're providing the kind of genomic support to that clinical research. So we're a core facility providing that sample processing, that's DNA and RNA extractions, biobanking of samples, and then genomic analysis. And we use lots of the Illumina platforms, such as the iScan for doing genotyping and methylation arrays. And we have the ISARIC, NextSeq 550, and the NextSeq 2000 sequencing platforms. So we're a core facility working on a wide range of projects, but that all stopped at the end of March with the start of the pandemic. And there we swiveled to doing COVID-19 work. So there are a number of NIHR urgent priority studies for hospital patients. This tweet here was released last week celebrating 100,000 people recruited to these high priority studies within the UK. We are working on 2 of them. The first one is the clinical characterization protocol on the top there, which has just passed 60,000 people recruited. And then the bottom study called GenOMICC, which has just passed 3,000 people recruited. And I'll talk about these in a little bit more detail. Now, I put this slide on here really to recognize the, I think, the amazing work and effort that has gone in from the clinical research community in the UK in such a short time, but mainly to recognize those volunteers who have volunteered for the clinical research, and without them, this wouldn't have been possible. And many of these people would have been very ill, and many sadly died. And so thank you to them and their relatives. So the first study we've been working on is called GenomiC. That stands for the Genetics of Mortality in Clinical Care. And this is led from Edinburgh by a clinician, Dr. Kenny Bailey. And we've been working on this project since 2016. It's a genetic study to look at emerging infections, mainly with the idea of things like a next round of flu pandemic, but also things like SARS and MERS. And so this was all in place before SARS-CoV-2, and we were able to use this structure to recruit patients and do the genomics on their DNA. And so it really ramped up at the start of the year, and we now have 204 intensive care units throughout the UK enrolled, and that covers about 5,000 intensive care beds. And the aim of this study is really to try and get every intubated patient, those most ill patients, recruited. And the aim is to try and get about 10,000 patients recruited. The second project we're working on is called ISARIC 4C. So this stands for the Coronavirus Clinical Characterization Consortium, which is really about getting data to answer basic questions about the pandemic using a set protocol. So things like who gets ill, how long are they infectious for, what are those kind of comorbidities. And it's a very pragmatic approach. It's really there to try and get people to get data from patients who are hospitalised, and they've successfully done that for over 60,000 patients now. And the idea is that for those ICUs that have the ability, that they can step up and start doing some sampling and get some single samples from patients. And as they get they have more time, they can then go on to tier 2 samples, which is serial sampling from 300 patients on days 1, 3, 9 and 28. And it's pragmatic because if the ICU department gets busy with more patients, they can drop down back to tier 1 and just do one set of sampling, or even go back to just data collection. The data collection being the most important thing for this study. Okay, so this has been very successful, and this paper was published in BMJ last month on the first 20,000 patients who had gone through the characterization protocol. This is led by Anne-Marie Doherty here in Edinburgh. And what it shows is on the top left that the disease really affects older people. And you can see also that it is more predominant in men. That's the left-hand side of that graph. And below that, protocol has shown those kind of matched symptoms, such as fever and the cough. And this is giving information on who should self-isolate. And then on the right-hand side of that graph then is those comorbidities. So things like heart disease, to kind of inform who should be shielding. So this is a very static data set in the paper last month. But every week, this data is being updated on the web page on the ISARIC4C net that you see there on the bottom right. And you can then investigate all 60,000 protocol reports there online. Sample collection is a huge task, and this is the kind of samples that have been collected on these timelines. You can see that there is serum, plasma, nose and throat swabs. All these samples are collected from the patient and sent from the different ICUs to the specialist labs in Liverpool and Glasgow, who are collecting all the samples in and then redistributing across the UK to specialists who have different interests in this disease. So my lab is getting blood samples for DNA and RNA extractions, and then we're doing the genomics on those. Other sample types are going to different labs. And this quote in the bottom, I think, is really interesting, that their aim is really to hand out as many of these samples as possible. So they're not intending to sit on a biobank, that they're trying to use as many of these as part of this crisis as possible. And I think that's a really strong statement of intent. But why are we doing this? Well, there's lots of variability in outcomes of people with infectious diseases. Some of that will be genetic. So in previous work we've done with Kenny Bailey, we were during the H1N1 pandemic. We were doing GWAS on patients who were ill in hospital, and that GWAS were able to identify IFITM3 as one of the genes involved, and that led to this paper where there was also some nice functional work. And in the recent pandemic, there's just a paper released last week in New England Journal of Medicine, likewise doing a genome-wide association study showing hits on chromosome 3 and chromosome 9 in the ABO blood region. Okay, so what are we doing for these studies? So they are quite closely linked, GenOMICC and ISARIC4C. We are getting those DNA samples into the labs. That's blood samples for GenOMICC and cell pellets for ISARIC4C. We're doing the extractions within my lab, and then we're doing genotyping on the Illumina global screening array, which is looking at about 730,000 SNPs. And then also what we're doing is taking an aliquot of that DNA, and we're sending that down to the Illumina labs in Hinxton, down in Cambridge, through Genomics England, and they're getting whole genome sequence down there. And the aim is then to do a GWAS and sequence the whole genomes of about 10,000 people. For the RNA, these are smaller numbers, looking at about 1,000 single time point samples and another 300 serial multiple day sampling. For these, we're doing the collection is either in the Tempus or PaxGene blood tubes. We're doing the RNA extractions, and then we're sequencing the messenger RNA, and we're using NebNext Ultra 2 kits, and then we're running those on either our NextSeq 550 or NextSeq 2000 with the aim of getting about 50 million reads per sample. So as part of this study, we've just got the NextSeq 2000. We were very excited in January when this platform was announced. It's a really good fit for my lab in the size of and throughput lab we are. We had planned to buy it before the pandemic, but we really needed it to get this RNA sequencing completed. And so here's a picture of the NextSeq that we managed to buy and get delivered in April. And I'd really like to thank Illumina here for being able to deliver, install and train us to use this platform. It's really helped us get the RNA sequencing completed for this study. Okay, so where are we now? So you can see from this graph that the blue line is the samples that we've received for the genomic part of the study, and the orange line is the DNA extractions. And you can see that we've had a busy time doing DNA extractions, but managed to keep up with samples coming into the lab. That we've up to date, we've now received just over 3,000 blood samples and have extracted just over 3,000 samples as well. We've genotyped just over 2,600 samples, and so you can see that's a really quick and cheap way of getting data immediately out of this study. We've sent about 500 samples down to Illumina for whole genome sequencing, and the plan is to get out to 1,000 by the end of this month, end of June, and with the expectation that those 1,000 whole genome sequences will be released to the research community at the end of July. On top of that, we've extracted RNA from over 500 samples and managed to sequence 240 of those. So I think it's been a fantastic effort from my team. I think it's too early to kind of say anything about the results yet, but I think we're really excited about this data set. So this has been a huge project. There are lots of people to thank, but I particularly like to thank Kenny Bailey for believing in my team and supporting us through this, to Andy and Wilner in Edinburgh for their support as well. But mostly I'd like to thank the outbreak labs in Liverpool and Glasgow, who've done a huge amount of work getting all these samples in and passing those out. Much underrated and very difficult work under very difficult circumstances. But mostly I'd like to thank my team, who've done a huge amount of work and have been very flexible and have done a fantastic job. And I know how proud they've all been to have contributed to this work. Thank you.